Good day. Welcome back to another revision stream. So, we have our mocks in exactly a week, or I guess now 6 days, because it's Wednesday evening. So, there are a few topics, I'm going to write them on the board, that I need to revise, and a few topics that I'm very confident with. So we'll start off, we'll do like a kind of ranking, okay? We got okay. So the topics that I personally am very confident on is the biological molecules topic. I love this topic a lot. I find it very easy. There's a lot of things I could ask you about it, so I'm happy with this one. Also very happy with the immunology topic. At some point I was pretty, here with it, but I think I've revised it in school a bit. Maybe we go over it 1 or 2 more times, then we'll be golden. Like, there's a few things like monoclonal antibodies we could go over maybe, or something to do with that. Mainly just exam questions. And I also just think the cells topic in general, we're pretty confident with that. But again, there's parts of that topic which can kind of expand into that. Like, I can't really think off the top of my head, but I know things like mitosis might need to go back over. The topics that I'm, not confident with is going to be things like meiosis or just genetics. I've never been really confident with this topic to begin with, but I think we're slowly getting there, so I'll pop it like that. And I think mass transport is one which we're doing today. That's going right in the middle. And digestion and absorption is going right there. So right now, the topics that we're going to go over today are mass transport. In school, we're going to go over these two. So that saves me plenty of streams to go over mass transport, plenty of streams to go over digestion, and we'll take 1 or 2 streams as well to go back through these. So that's a plan. That's the plan for the streams. That's what we'll be doing for the next week until this exam is over and done with. So, let's get a move on then. Let's start by looking at the structure of hemoglobin. Reason it's called hemoglobin is because it's a globular protein and it contains heme groups. It's comprised of heme groups. It's a quaternary protein. I've been pronouncing that wrong this whole time. It's called quaternary, I'm pretty sure. And what that means is it's multiple polypeptides joined together. So it's bonded together by, I'm pretty sure, disulfide bridges. Like so. And each of these individual proteins is comprised of a heme group, which is, I think, 2 iron ions. Remember, with these streams, I always just dump what I know first before I check if I'm right. So that's the structure. Now, each of these iron ions is able to bond with, I think it's, each heme group can bond with 1 or 2. So, keep that in mind, each heme group combined with 1 oxygen. There is the dissociation curve and the Bohr effect. That's what we got to talk about. Dissociation curve is where, to begin with, it is pretty difficult for the oxygen to be absorbed into the hemoglobin, but then eventually a conformational shift occurs, which makes it easier. So there is a slight shift in the tertiary structure. That's the conformational shift. And the structure changes, which makes it easier for O2 to associate. But then, eventually, when all the heme groups are saturated, obviously the chances of an O2 molecule, an oxygen molecule, binding with that specific heme group are a lot more difficult. So as a result, it's more difficult. So that's the dissociation curve. Very easy, very straightforward. The Bohr effect is where during the reaction of respiration, CO2 is produced, and inside the cell this forms carbonic acid. Acid is acidic, and that will increase the pH. And what that means for our molecule is it will become slightly denatured, which means that it will dissociate oxygen more readily. So, hemoglobin. We've talked about the dissociation curve, talked about how the Bohr effect works. Let's look at a red blood cell. Some properties of this is that it's a flat, biconcave disc. It has no nuclei to increase the surface area. So that's the main thing. A large surface area to volume ratio. And no nuclei, which means obviously it's maximizing the amount of space for the heme group to be there. Have we talked about everything? Let's quickly talk about surface area to volume ratio, because I don't recall this one too often. Surface area to volume ratio is this idea that smaller organisms, compared to bigger organisms, smaller organisms smaller organisms retain temperature, I can't word it properly. They retain, temperature less. They're unable to retain things like temperature. It's an incredibly small diffusion pathway compared to large organisms where they have very large diffusion pathways. So I guess the main point of surface area to volume ratio is this idea that the smaller organisms have less diffusion pathway than the big ones. Okay. So we will leave it there and go check with the book to see what we need to talk about again. Because there is gas exchange, which we're doing today. So what we've done today, we'll go through surface area to volume ratio. We'll go through blood vessels, and we'll go through gas exchange. Then next time we'll go through heart, water transport, things like that. Because I always forget water transport is a thing. Okay. If I can find the questions. Exchange with the environment. That's where we're at. Surface area to volume ratio. Very simply, all organisms must constantly exchange substances. This isn't a very helpful book. Large animals compensate by having... So animals that are large have a smaller surface area to volume ratio, we know that. They compensate that by having features which allow them to do things like retaining temperature. So for example, things like foxes might have things like pointed ears, which would increase their surface area, meaning that they lose temperature faster. So it's this idea, one point is temperature. Smaller organisms lose temperature more readily. Large organisms don't. And that applies to not only organisms, but just their adaptations. The larger the surface area of something, the more it's going to lose its temperature. And surface area to volume ratio is always going to be put of ratio. You pretty much get given the formula for this in an exam, so yeah. Where's hemoglobin at? Where is hemoglobin? It's going to be in the heart, isn't it? See, we're actually jumping between two at once here, two topics. Okay, hemoglobin is a red pigment that transports around the body. It's a soluble protein quaternary structure. 4 heme groups in 1 hemoglobin. Right, hold on, I have misinterpreted here, I think. 4 heme groups in 1 hemoglobin molecule can combine with 1 oxygen. So I was wrong. It's just 1 heme group per. But we were right about this. We were right about the fact that each heme group can only temporarily associate with 1 oxygen molecule. Cool. When oxygen concentration is low, it disassociates, it splits up, yeah. So, just basic terminology there. Association, disassociation, this idea that it associates with oxygen, and then when it releases it, it disassociates. Very simple. We've talked about the disassociation curve. Let's check if that's right. If each molecule, okay. The percentage saturation of hemoglobin increases. Yeah, so I've drawn the curve differently here, but it's the exact same gimmick. This is how it really should have been drawn. Like that. But I've drawn it to represent how much it can associate in a way. I know what I mean in my head, but I've drawn it differently to how they would want you to draw it. So the conformational shift we've talked about, and when the hemoglobin is saturated, when the heme group is saturated, it becomes more difficult for it to associate with oxygen. So it's an S-shaped curve, yep. Oh, there is another curve. Okay. So there are 2 S-shaped curves, apparently. Where am I going to draw this at? Bollocks. I'll draw it here. Hold on one sec. Okay. We're good, I think. I might have just ended the stream. The S-shaped curve, which I always kind of struggle to draw, that is not an S at all, but who cares? The S-shaped curve is this idea that when partial pressure changes, it becomes easier or more difficult to associate with oxygen. So if you increase the concentration of oxygen versus decrease, it is still going to eventually reach the top, but it's going to take longer to do so, or shorter. So in areas where oxygen is less readily available, it'll take longer. So yeah, when there's less CO2, it happens faster. There's a, yeah. Curve shifts to the right in the presence of CO2. Okay. Right, I think that we have talked a lot about this. I think that that's everything to do with hemoglobin and red blood cells as a whole. But to quickly recap, hemoglobin is a globular protein with a quaternary structure. Each polypeptide has a heme group which can associate with one oxygen molecule. And all in all, based on things like CO2 concentration, O2 concentration, partial pressure, it will become easier or more difficult for the oxygen to readily disassociate, sorry. The structure of a red blood cell, we know this already, it's very basic. Large surface area to volume ratio, non-nuclei, etc., which will make it more adapted to its purpose of having hemoglobin in. Bohr effect, this idea that carbonic acid denatures the polypeptide slightly, and obviously the dissociation curve, meaning that it does become slightly easier to associate with oxygen the more there is up until a certain point. Right. What are we doing next? We could talk about insects, humans, and fish gas exchange. I don't know. I don't know. We literally have two choices. Do we talk about... Bollocks, let's just get it over and done with, shall we? Because I don't like this topic at all. I don't. I'm putting myself off doing it. Right. Should we start with humans, because they're the most annoying one to revise. I always forget how to do it. It's about the intercostal muscles and things like that. Inspiration and expiration. So... Cool. Right. Let's do this. So I'm gonna just try and recall as much as I can off the top of my head, and then I'll check the textbook and kind of re-add. Probably would be worth checking a mark scheme here, because mark schemes can be very picky about the words you use. So I'll do that once I end the stream. Why is this pen so shit? Have I lost a pen? Bro. I am not crazy, I've lost a pen. Are both of these orange? No, that one's... What the hell? What? Right, I'm actually going crazy. I've lost a pen. Yeah, okay. I'm actually gone mental. Okay. Inspiration. Let's talk about it. So, it's this idea that you have your internal... external intercostal muscles. External... these are going to, I'm going to guess, contract. If you're breathing, hold on, inhaling, it gets pushed outwards. Like, I remember that's one of the things, is the diaphragm is pushed outwards. Or like the rib... Oh, yeah, fuck it, I'm checking the mark scheme. I'm just going to start... Yeah, the muscles in the diaphragm and the external and intercostal muscles. Okay. So it's the diaphragm muscles. Right. I'm making this like a fucking table now. I'm not spelling diaphragm the right way because I'm illiterate. Diaphragm, external, intercostal muscles. Cool. Okay, so This makes it easier, I think. This makes it easier. So, they do not explain it here very well. What? Breathing in is always an active process. It is brought about by two sets of muscles. Both of these muscle groups contract. Okay. I'm guessing that the contraction of the diaphragm will cause it to be pulled down. Pulls the rib cage up and out. I remember this now. Again, the annoying thing with this biology topic is it really does seem very systematic in the way that it forces you to memorize like mark schemes. Because I can't lie, that's about 80% of the way I revise stuff now is just looking at mark schemes to try and memorize the exact terminology, because you will get opped on if you don't know the exact terminology. It's really weird. But that seems about right. The diaphragm contracts, which pulls the muscles down. The external intercostal muscles contract, which pulls the rib cage up and out. Obviously, that's going to increase the volume in your thorax. I remember this bit. It increases volume in thorax, which creates a pressure gradient, because there is now... Is it a pressure gradient, I'm assuming? Because then air will diffuse in. So there's a greater pressure? No, there is... what is it? It increases the volume. Yeah, it decreases the pressure because it's moving down a gradient. My bad. So there's less pressure because you've just increased the volume of it, which means that air is going to diffuse in from the outside, the air, the atmosphere. So it increases the volume. That's going to make a pressure gradient. Cool. And then meanwhile, with expiration, I'm guessing it's the opposite. They're going to relax, which decreases the volume, and as a result of that, air is going to be pushed out because there is a higher pressure in the thorax than there is in the air. Am I correct? Also, interesting thing I didn't know, that is an active process, this is a passive process. I guess it makes sense because your muscles are contracting here, which requires energy. This is a passive process because your muscles are relaxing. Possible mark point there, because I feel like they could ask you something about that, no? I feel like they could be scummy and be like, which of these processes requires ATP? Something like that. I feel like they could easily ask you a scummy question about that. Or they could even be like, which process involves the contraction of these muscles that is active, and that would make people panic. Right, there is a... Oh my god, I'm actually such a tit. There is a diagram right here which would have cleared all of this up. Right. External intercostal muscles contract, raising the rib cage and increasing volume in thorax. Diaphragm muscles contract, lowering the diaphragm, increasing the volume. So both of these increase the volume, yeah? I already knew that, but just to reiterate. Breathing out, the external intercostal muscles relax, decreasing volume. Diaphragm muscles relax. So in other words, they just go back to their normal position here. Intercostal muscles contract. Oh, so during exercise, that is one thing I remember the teacher mentioning, that I was wondering when this would be brought up. The internal intercostal muscles will contract slightly to lower the volume, but usually it's only during exercise. I will have to check whether the mark scheme would allow you to mention that or not. Because again, I'm forced to think like a mark scheme during these streams. I don't know whether they would want you to talk about that. I have no idea. I could put that in an exam and get it wrong or get it right. It's a gamble. So I have no idea what they would want off me though. But there we go. There's a couple mini questions. I could quickly do those. Explain why the body obtains less oxygen per breath at high altitude than at sea level. I'm guessing this is to do with partial pressure. I don't know how you would word that though. Yeah, air pressure falls to a level below the pressure in the lung capillaries. Okay, interesting. Oh, this is like reading comprehension. I was wondering why I didn't know what any of those words meant. I'm not dense, I promise. Should we quickly do blood vessels, and then we'll move on to insects and fish next time? That is decent, though. I can remember this now. See, a lot of processes in biology aren't that difficult. It's just about remembering, very specific niche terminology. And a lot of my energy is not diverted to it, so that's why. We have the veins. Have I spelled veins wrong? No, that's not how you spell vein. I'm losing the plot. Okay. Artery and capillary. I am already remembering a couple of things flitting back here. There are different types of, arteries and veins, I don't recall, that link to capillaries, right? I remember this being a thing, this kind of weird flowy thing where it kind of links. I don't know, I remember something like that, but anyways, we'll see what I can remember and then we'll look at the book. So, veins have arteries are going to have, is it a narrower lumen because it's made of elastic and muscle tissue? Arteries also have the ability to contract, I believe. Veins contain valves to prevent backflow. They have a wider lumen. Valves prevent backflow. These have a narrower lumen, elastic, and muscle tissue, and they can contract. And capillaries are quite literally just there for the crack. I don't know how to describe them, so let's have a look. Yeah, wall made of elastic tissue and smooth muscle. Oh, there's the endothelium as well. That's the tiny, tiny, tiny part that you don't learn at GCSE, which is just like that. Yeah, cool. Endothelium. So, the arteries are lined with a layer of flat, smooth muscle cells. The rest of the wall of the aorta and other large arteries is made up with a thick layer of elastic tissue, fibrous tissue, smooth muscle. It is stretched by the pressure produced by the ventricles. It maintains a high pressure. The expansion and recoil of the artery walls helps the smooth surges in blood flow. Okay, so I remember there being an exam question where it was on about how veins and arteries, this idea that they help maintain pressure, because of their ability to, oh my god, because of their ability to contract and relax via the smooth muscle. So that is an interesting point. Oh, it's arterioles and venules. That's this idea. So you have like the main artery and like the main vein, and you have this idea that it will branch off into arterioles and venules, and then these will branch off into capillaries to kind of link the two together in this gradient. So it narrows the, they can restrict blood flow too by contracting. So, hold on, there's two different properties, right? No, we're on about arterioles now. Capillaries act as an exchange surface between gases and other solutes. They are made up of a single layer, one cell thick we know about. They are one cell thick because of that. Yeah, okay. And this idea of blood pressure. And tissue fluid. Yes, okay, that is something. So, I think we'll leave things there. Just gonna write this down for next time. This idea of the cardiac cycle. So that's all we need to do for the rest of this topic. Talking about fish and insects and their gas exchange, talking about tissue fluid, going over arterioles and venules, and then the structure of the heart, and then the cardiac cycle. So I'm pretty happy with that. We went over hemoglobin again, went over blood vessels quickly, and revised a decent bit about inhalation and things like that. So I'm happy with wrapping things up there. I'll see you guys in probably a day.